Good afternoon to all of you. The next topic that I'm going to discuss with you in the next 20 minutes is the molecular diagnosis of chronic endometritis. First, as I did before, I have to disclose that I am the founder and head of the Scientific Advisory Board of Igenomics, a biotech company working from the bench to the bedside, and some of the topics that I'm going to present here has been translated as a product, as a test, and therefore you have to know this situation. So chronic endometritis is the persistent inflammation of the endometrial lining, mainly initially caused by microbial pathogens. That's the description. So any type of inflammation, any type of inflammatory situation, it is causing by a pathogen. In this case, the most common bacterial pathogens involved in chronic endometritis, you have it in the slide. They are E. coli, Klebsiella, Streptococcus, Enterococcus, Staphylococcus, Mycoplasma, Ureaplasma, and Chlamydia. All of them has been linked directly to this inflammation of the endometrium. The estimated prevalence of this pathology is between 1 to 20% in the general population and between 3 and 40% in infertile patients. You have in the slide some of the papers in which we work on that in order to get this estimated prevalence. The issue is that this is not a pathology that we can just subestimate. It exists. The issue is that we are not able to really diagnose properly, but it's there, and most frequently in our most difficult patients. Usually, as mentioned, it's asymptomatic, so it will go without diagnosis and treatment. Patients may suffer from inspecific pathologies such as metrorrhagia, cervical discharge, or pelvic pain, which is quite of, is not like something very specific. So because of that, we will not be sure what is going on there. And we know that chronic endometritis influences reproductive function. It has been linked, it has been associated with recurrent miscarriage in around 50%. It has been linked and has been shown that the prevalence is about between 30 and 60% in repeated implantation failure, and also related to preterm birth and the fact that delivery starts before it should. So you have the reference behind this, in which we have been gaining this knowledge. So this is not a minute issue, that the fact that this infection may follow during pregnancy, affecting everything that comes after that. But not only in pregnancy, but also the fact that chronic endometritis affect the reproductive outcome in IVF. And you have here this systematic review and meta-analysis in which clearly, and Ettore Ciccinelli is one of the main authors contributing to this, the knowledge that we have on chronic endometritis. As you see, it is a clear deleterious effect of chronic endometritis in terms of reproductive outcome, as you can see in the slide and in the paper published just recently. Now, the issue, the battle here is that this pathology is inspecific clinically, but diagnostically wise, it's quite of difficult to diagnose. First of all, it's not detectable by ultrasound. And there is 3 classical methods that has been used in order to diagnose it. First is histology, which still is the gold standard with the presence of plasma cells, plasmocytes, that they are coupled with immunohistochemistry for CD138. And as you will see, it has pros and cons. The second is hysteroscopy, the introduction of the scope in the uterine cavity and to see images that they can really guide us to say there is an infection there and therefore there is endometritis. And third is the use of microbial culture, which is stable, and culture them in order to see the pathogen there. Each of them has pros and has cons. The histology, as mentioned, is still the gold standard. We aim to see those cells that are plasmocytes or plasma cells. They are coming from B cells, and also we can couple with CD138. Now, the problem is this a completely subjective criteria. There is no criteria in terms of how many plasma cells per field. There are different protocols from different institutions. Second, B cells are frequently found in the endometrium during the secretory phase, and this doesn't mean that there is an infection. And third, the most important, no identification of pathogens exist. We just assume that the presence of a specific pathogen, as we just said in the first slide, was the presence of those plasmocytes. But this is not a clear-cut association. The second technique is hysteroscopy. We are all aware of what hysteroscopy is, and we are all aware of the different diagnostic criteria with different images that has been linked to the existence of chronic endometritis. And you can see this in the Delphi study in a very nice and rich description of those images, pretending that with that, just say, okay, this is an endometritis here. But again, the cons of this is a subjective technique, depends very much on the observer, which means depends on the experience of the observer, and also depends on the phase of the menstrual cycle, and does not allow at all to identify a specific pathogens that should be upstream as the cause of chronic endometritis. And third, the microbial culture, which is the obvious thing to do, which is let's take a sample and isolate those microorganisms that they are present. And as you can see, they are able to identify some of them, these 5 that you have here, but those that are the most important one, for instance, Gardnerella, for instance, Neisseria, they cannot be basically culturable. So the most important cons that this technique has is that the fact that 20 to 60% of bacteria, they cannot be cultured. And that's the difference, and that's why the introduction of next generation sequencing makes a difference in the identification of the bacteria, because with the 16S ribosomal RNA, we are able to identify the existence of this bacteria. This is the main issue of microbial culture, in addition to the individual isolation of pathogens. Keep in mind that every time that we culture a sample, we are enriching this particular media to grow those bacteria. It doesn't mean that any other bacteria can grow, and considering that the amount of them that they will pass without non-culture, this technique is not anymore used now as a gold standard to identify what is there. Now, altogether, makes that this pathology is today flowing as a spectrum of estimations and subjectivity. And at the end, patients, they are being treated with broad spectrum antibiotics, which we do not know exactly what they are doing, not only in terms of treating the pathology, but also in terms of what is going to happen afterwards. Because keep in mind that, as you will see, that the microbiome is an ecosystem. And if you just treat them indiscriminately, pretending treating one pathogen, you are just causing that disbalance that may produce even worse effects that we were planning to do with our. Now, let's go one step back. And I just want to inform you about that there is a microbiota continuum in the female tract. It's not like the existence of Gardnerella in the endometrium means that there is an isolated infection of Gardnerella. Is the point that there is a disbalance. We know, and has been fully demonstrated, that the microbiome, the presence of microbes in the reproductive genital tract, is a continuum, starting from the vagina, where almost everything is lactobacillus. I'm referring to the physiological microbiome, the cervix, the uterine cavity, in which lactobacillus are present, but other pathogens may exist, then the tubes, in which lactobacillus are not there, and there is another type of species, and finally, the peritoneum of the microbiota of the peritoneum cavity, in which now the landscape has changed. So this is the physiological landscape of the microbiome in the reproductive tract. And we know that the endometrial microbiome exists, and we know that if there is a disbalance in this endometrial microbiome, it will affect the reproductive outcome. And we showed this a long time ago, 4 years ago, in this paper in the American Journal, in which by taking endometrial fluid and endometrial samples and follow the patients, we know that those that achieve live birth, the profile is completely different than the ones that have no pregnancy or clinical miscarriage. Blue is lactobacillus, the good one, and the other colors are the other pathogens. Some of them, they are also implicated in chronic endometritis. So this is the situation. And now let's go specifically to chronic endometritis. What we did in this paper in the American Journal is we tried to understand how consistent are the three methods of the diagnosis of diagnostic of chronic endometritis, and how molecular microbiology may play a role, as it is playing in any other field in medicine, in improving our detection capability and our treatment capability of chronic endometritis. So what we did for that is just taking the endometrial tissue, and instead of using these three techniques that I already mentioned, we went also to understand what happens in the bacterial DNA by either a simple RT-PCR of these 8 pathogens causing endometritis, or just going to metagenomics using next generation sequencing, analyzing the very same pathogens using these 2 different technologies. So this was the idea for the molecular assessment of chronic endometritis. This is the flow of the paper already published 2 years ago, in which the samples were sent to us blindly by our colleague Ettore Ciccinelli from Italy. Those were patients that they have been investigated by the 3 classical methods. We just received the paraffin, the endometrial samples in paraffin, and this was the samples analyzed by the 3 classical methods. And then we distinguished between those that they were concordant between them and those that they were discordant. And then we did all the comparison with microbiological, with the molecular analysis of these endometrial samples using the system that I mentioned to you. Well, the 2 most important messages that I will convey to you is that when we analyze what is the accuracy of the 3 classical methods, histology, hysteroscopy, and microbial culture, in all of them, we could see only that they coincide in 20% of patients. Only 20%, 13 out of 65, the diagnosed were concordant. In the 80% of patients that for some reasons by histology, they were categorized with chronic endometritis in blue, in yellow. Some of them, they were not found in others. As you can see, hysteroscopy was the method that diagnosed most of the chronic endometritis, and microbial culture, the method that diagnosed less of them. And the other message to convey to you, and I will try to show you the data, is that when we applied molecular microbiology to those samples that they were concordant to the classical methods, 13, about 77% of them, we have an accuracy of 77% of them, indicating what is the value of this technique on that. We have, as you can see here, that the histology, stereoscopy, microbial culture, and RT-PCR were concordant. And we have patients in which we have discordance, negative for different histology, stereoscopy, positive for microbial culture, and then we have a positive for microbiological analysis. And as you can see that, this is the assessment of chronic endometritis by NGS compared to the classical methods. Once all the classical methods say is negative, yes, we are with was negative, and in some cases there was positive, and we find no pathogens, and in this case, patient 26 again. So this is just to show you what it's the treatment and what has been the agreement between the three techniques itself. We even were asked by the reviewers to be able to discriminate between positive and negative cases of chronic endometritis using this molecular test. And we use as a control patients that they were negative, they were found negative, and those patients that they have been positive for chronic endometritis, they have, and now they are negative. So we were able also to find that. And this is the final table from that. So this is about the sensitivity, specificity, and accuracy of the classical diagnostic methods of chronic endometritis, histology, stereoscopy, microbial culture, and then all of them three together and see what is the concordancy in terms of molecular microbiology. There is another paper from an independent group showing similar results as we have already mentioned, in which patient with the microbiota with patient with chronic endometritis, as you can see, it's different from those with non-chronic endometritis. This is lactobacillus, which is higher than those patients that they did not suffer chronic endometritis versus those that they suffer chronic endometritis. So in conclusions, what I mean from this lecture to bring to you is that the molecular microbiology effectively detects bacterial DNA, chronic endometritis causing pathogens in endometrial samples, including, very important, non-cultivable bacteria. The accuracy of the molecular diagnosis of chronic endometritis is similar to histology, hysteroscopy, and microbial culture together, overcoming the bias of using any one of them alone. And finally, the point that molecular methods must be considered for the diagnosis of chronic endometritis in asymptomatic infertile patients to improve current decision-making of this invisible endometrial pathology. So this is just to add to our knowledge, just a different view, a different angle on objective diagnosis of a pathology that nowadays still is in the cloud. It's not inflammation. It should have an infection, a microbial infection, and the best way to really determine its presence is by detecting the DNA of the bacteria causing this infection. And with that, I just want to show you the face of the team, of the group that have done the work, leading by Inmaculada Moreno, who is the principal investigator of the endometrial microbiome project in my lab. Thank you very much for your attention.